Hello and welcome back to educator.com. Thanks for joining me. This is going to be our last lesson in this molecular biology class. And what I wanted to do is talk about a few subjects that seem to be kind of glazed over, if it even touched upon at all, in normal college classes because they often run out of time. You know, there's certain information that needs to be covered, and while this is very interesting stuff, it's not of the utmost importance, and it's really hard to lecture on. So in no way am I going deep into this. I'm just giving a surface level view and trying to just get your mind working, trying to get, you know, the topics into your head and just have you thinking about it a little bit. So we're going to talk about genome editing, synthetic biology, and stem cell biology. And we're going to talk about the ethical aspects of each of these, as well as just ethics of science in general. Many of you who end up going to graduate school will actually have to take seminars in research ethics. And so this is something that maybe will just be a starting point for you guys. For any of you who work in a lab, you know, this is going to be something that you always have to keep in mind, and it's always something that's going to surround your projects, and it's good. We need to think about this. So I'm going to do my job of introducing it to you, and hopefully you take this upon yourselves to really think about these aspects. So genome editing, let's start with that. So first of all, what is it? Okay, it is genetic engineering, in which DNA is either inserted, replaced, or removed from a genome using artificially engineered nucleases. Remember, nucleases are the enzymes that cut up nucleic acids, DNA and RNA. So how do they work? Double-strand breaks get introduced by the action of the nucleases at desired locations in the genome. These double-strand breaks then just get repaired by the normal mechanisms that we've talked about in this molecular biology class of homologous recombination or of non-homologous end joining. Now, there are 4 families of engineered nucleases that are in use for these processes today. We have zinc finger nucleases, we have talons, we have meganucleases, and we have the CRISPR-Cas9 system, or just the CRISPR system. Meganucleases, they usually cut on average once per genome. But I'm going to focus on the CRISPR-Cas9 system because this is actually a really interesting system, and it's in use, and really, really cool things can be done with this system. So for genome editing, I'm just going to focus on this one. So here's our example. Here's our CRISPR-Cas9 system. So what is CRISPR? It stands for clustered regularly interspaced short palindromic repeats. And Cas9, so the Cas proteins, that is CRISPR associated protein. And in this case, Cas9, that's CRISPR associated protein 9. Okay, so what is this? Okay, so basically what CRISPR is a very, very, let's say, primitive adaptive immune system for bacteria. Okay, so it is their, so if we're looking here, this is a cell membrane of a bacteria, it is their way of defending against a bacteriophage, which is a virus that infects bacteria. Okay, so this is a primitive adaptive immune system for our prokaryotes, right, our bacteria. So this CRISPR, okay, it's clustered regularly interspaced short palindromic repeats, okay? And what it does is you have repetitions of base sequences, okay, in here, followed by spacer DNA. Okay, so such as this. Okay, these are spacer DNAs. Okay. Those spacers will recognize and cut, so act like an endonuclease, cut up invading viral DNA. Okay? And it'll cut, it's almost similar to RNA interference, but it's a different system for doing that. Okay, so how does this work? So first, the virus, right, latches onto the bacteria, injects its DNA. Okay, so we have a double-stranded viral DNA. It's really, it can be simplified down to a 4-step process. So the invading viral DNA, double-stranded DNA, or if it's an RNA virus, same thing could happen, is cut into small fragments. Okay, and from those fragments, you end up creating a novel spacer. Okay, so this is some DNA sequence, and this is basically a way that links this is linked to this specific piece of DNA. So the invading DNA or RNA is cut into small fragments. So that's step 2, or step 1. Step 2, a copy of that invading DNA or RNA is integrated into the CRISPR locus as a spacer. Okay, so this one, okay, this corresponded to a different piece of viral DNA, and this one to a different one, and to a different one, and to a different one. Okay, it's an adaptive immunity, meaning it's seen that before, it keeps a piece of it, so if it ever sees that DNA again, it'll be able to break that down and silence it. Okay, so step 2, right? You make a copy of it, you put it into that CRISPR locus as a spacer. Third step, the CRISPR RNA, which is called a cRNA, so CRISPR RNA, okay, they get transcribed. So, okay, we'll call this, here's step, this is step 1, this is step 2, this is step 3. Okay, so the CRISPR RNAs are transcribed from the CRISPR locus. And then step 4, we have the cRNAs, the CRISPR RNAs, being incorporated into the CRISPR-Cas complex. And then 5, it will target viral DNA, so invading viral DNA, and it will degrade that. So if you want to call that, this is 6. Okay? And the cRNA will provide specificity. So it will act only, okay, this cRNA produced from, these specific spacers will specifically act on what it's already seen, okay? And researchers can utilize this as the cRNA, the CRISPR RNA, providing specificity for such things as, let's say, gene editing for medical purposes. So you can silence something, let's say if you have an overactive gene that's causing a disease, you can use the CRISPR-Cas9 complex, okay, to utilize that, right? So you could silence that. Now, there's plenty more in-depth detail to go into this that I'm not going to go into, but I recommend that you try to look this up. This is really, really cool, fairly cutting-edge techniques being used here. Now, gene therapy, right, specifically, that's our delivery of nucleic acids into a patient's cell for treatment of a disease, okay? You could either be adding a gene, you could be knocking out a gene, you could be replacing a gene, okay, any of that. Now, the polymers of the nucleic acids are either expressed as proteins and therefore interfere with protein expression, or directly correct genetic mutations by replacing the actual DNA sequence. So the DNA that encodes a functional therapeutic gene will replace the mutated gene, and then therefore when it is transcribed and translated, you will get the wild type protein instead of the mutant protein that has been causing the disease. So gene therapy, the theory on it, was first published in 1972, and the first experiment approved by the FDA occurred in 1990, so about 18 years later. And 13 years after that, the first commercial gene therapy was approved for use as a cancer treatment. And the gene therapy, called Genome, was approved in China in 2003. Okay. So an example of a gene therapy, right here, we can see this is gene therapy using an adenovirus vector. An adenovirus is a specific type of virus. What it can do is you can manipulate the DNA inside the virus. The virus will infect a cell, okay, bring that DNA into the nucleus of the cell. That DNA, now being a good piece of DNA, right, not something we need to worry about of like viral DNA, but something that we plan on putting there that will be healthy for the cell, will then make the certain proteins that we want it to make and will help, if used properly, to treat that disease. Now, this is a little different from RNAi or from the CRISPR system in the fact that we have something that we always need to take into consideration when we involve a virus. Yes, it is. There are plenty of research on it and plenty of ways that it has been altered so that it should not be able to replicate and cause disease as the normal virus would. But that's always something as a scientist and as the public you keep in the back of your mind. But for the most part, it does its job without infecting, quote unquote, in a bad way, but infecting in the good way, getting in the cell, giving its DNA, which has the good DNA that we want. To make the protein of interest. So some ethical questions revolving around gene therapy is, there's always going to be problems with genetic engineering. Okay, maybe the protein that you wanted to be sent in there, maybe there's a mutation on the gene, maybe you didn't engineer the gene properly, or just maybe the ethical question of just revolving around genetic engineering itself, manipulating the genome, playing God, if you want to say it that way. Okay. Another question that always is going to surround gene therapy is the aspect of gene doping. Okay, meaning basically, if used improperly, you could almost use it as a performance-enhancing drug alternative, okay? So you can do gene therapy to make you bigger, faster, stronger, have more oxygen carrying capacity, something like that. So that's always a question that will be thrown around. Okay. All right, moving on to a different topic, synthetic biology. Now, synthetic biology is described as the design and manufacture of biological components that do not exist in nature. So there are many ways of looking at synthetic biology. I want to look at a specific one, and this is an example of synthetic biology at work. Now, in 2010, scientists at the J. Craig Venter Institute, if we remember, J. Craig Venter, he was the entrepreneur that was involved in pushing the private funded aspect of the human genome experiment. Okay, the Human Genome Project. So scientists at this institute that's been opened, ran by him, created the first synthetic cell. Okay, so what they did is they started with a bacteria, okay, called Mycoplasma capricolum. And what they did is they took out all of the DNA, okay, any genetic material in there, and replaced it by using a transformation technique with a brand new fully synthesized, okay, fully synthetic, fully brand new, right? DNA made from their typing on a keyboard, right? So they made it in a computer, made a bunch of DNA. Okay, you can get DNA made, and they inserted their computer-coded DNA into this completely empty cell. Okay, it's like a shell at this point. And they obviously tried it many times until one worked, and they finally found a sequence, okay, that was actually able to replicate the cell as if it were a normal cell. Okay, and in this case, they actually created a new species that they called Mycoplasma mycoides. Now this is huge. They were able to take a man-made coded DNA sequence and put it in a shell of a bacteria to make a living, replicable organism. I mean, this is a huge, absolutely huge event. But with such a huge event like that, there's always going to be ethical questions. Now, one of the big questions that surrounded this was, did they create new life? Or did they just manipulate life? And you know, you can have your own opinion on this. One of the rulings that I believe it went to the court system was that this wasn't actually the creation of a new life. However, this is something that will always be asked about. Okay. Some other ethical questions regarding synthetic biology is, you know, GMOs are an ethical debate in and of itself, but when you start being able to computer code genes and be able to put it into an organism, that brings up an entire new set of questions about GMOs. And finally, the fear of this synthetic bacteria that they made, right, does not exist in nature, wasn't intended to exist in nature, right? We made it exist. We as a human race made it exist. What is that going to, what type of effect will that have on the current ecosystem, the current living system? And can that bacteria infect other species, right? They took very, very good precautions to make it in a non-infective bacteria, non-virulent strain, but there are ways that it can mutate, or there are ways that maybe somebody else might not take as great of care, okay, or might use this for ill purposes, okay? So that's always going to be an ethics debate. All right, and now moving on to stem cell biology, and I know this is a big, hot topic. This has been ethically debated since the thoughts of stem cell biology came out. So what is stem cell biology? You're using stem cells to treat or prevent disease. Now, often you have your stem cells coming from the bone marrow, right? So when you talk about bone marrow transplants, like for a lot of times people with cancer, right, or multiple myeloma, right, one of the specific ones, you need to get bone marrow transplants, and that's because cancer, as well as the chemotherapy, ravages your quickly dividing cells, such as your B and T cells. Your B cells, your part of your immunity, that is found in your bone marrow. So you need bone marrow replacement. You have your what are called hematopoietic stem cells. These are stem cells that can turn into any type of white or red blood cell. Your B and T cells are a type of white blood cell. So this is very useful and a very common practice nowadays. But you know, some people do not agree with the fact of umbilical cord blood being utilized as a source of stem cells. And a huge contest or point of contention is the fact of actually using embryonic stem cells, so stem cells from fetuses or unborn fetuses. That's a huge point of contention with many groups, especially your religious focus groups, you know, as well as even some non-religious focus groups, and some scientists don't agree with it. Okay, so you can also have embryonic stem cells. You can also have what's called somatic cell nuclear transfer technique, but you need to start with some sort of a cell to begin with. Okay, The good thing about stem cell biology is that it can be used to treat many, many, many types of diseases, okay? We have leukemia and lymphoma, diabetes, injuries to the brain and spinal cord, deafness, anemia. Anemia is the low concentration of red blood cells, okay, which will then affect your oxygen carrying capacity. As well as stem cell biology can be used to treat organ and limb transplantation to help that go as well. So some of the ethical questions. Are you destroying life, right? If you're talking about using embryonic stem cells, are you actually destroying a life that should have been? Okay, and that's going to be a point of contention that people are going to have, a side versus the other. There aren't enough, a lot of these decisions on ethics, you try to use as much facts as possible, but really it's going to come down to belief systems and how you can manage that between, yourself and the people that you have a contention with. Okay? Another one is human cloning. If we're using, stem cells to make other humans, right? That's a huge point of contention. And as of right now, human cloning is not allowed. Will it be in the future? Maybe. We don't know. So, other than the ones that I had mentioned before, there are many other topics that are up for ethical debate. Some that I wanted to list here: genetically modified organisms, vaccines, right? The whole vaccines, people be thinking that they're correlated to autism, right? Based on a study published by a medical doctor who named Andrew Wakefield in The Lancet, who was actually paid by a competing vaccine company to falsify data and talk bad about the vaccines that are out on the market, which has spawned, even though he lost his medical license once it was found that he falsified his data. The paper was retracted, but a lot of people don't hear that part of it. They only hear the fact that the story was published years ago. Okay, so this is always going to be for debate, and a lot of people don't have the right facts to follow that one specifically. We have other topics to debate, like savior babies. A savior baby is when a couple has a child with a condition where they are dying because they lack, let's say, for example, they have a kidney disease and they need a kidney transplant. Sometimes a couple will have another baby that was basically meant to use that second baby as basically an organ farm for the treatment of the first child. So the second baby was born so that it can give a kidney transplant to the first child who was sick. Okay? There's always going to be ethical debate over that. There's ethical debate over genetic manipulation. So gene editing for health, you will have people on both sides, as well as the Build-A-Bear type baby. Okay, I want to make my baby blue eyes, 6'5", highly athletic and highly intelligent, right? Who's to stop somebody from picking all the best qualities to make their child just, like a freak of nature, right? Like, I want to make somebody with the, basketball skills and body type of LeBron James with the mind of Albert Einstein, right? It's something crazy like that. Prenatal genome sequencing for the same reason of being able to kind of do that Build-A-Bear baby. Genome sequencing and insurance companies. So if I sequence my genome because I want to know what diseases I'm susceptible for, and insurance companies are allowed to have that data, they might not offer me insurance at all, or might have an extremely high premiums based on my risk factors. Okay, so that's a huge debate. Cloning, always a big one, especially human cloning. Animal testing, big debate, right? you're always going to have groups like PETA and even other scientists telling you, that animal testing doesn't need to happen or does need to happen, right? So you do need to test on animals if you want to pass selected drugs through the FDA to get to the market. So like all the, say your Advil, your Tylenol, even all the way up through, let's say, your chemotherapies, they have all had to have been tested on animals before they are allowed to go into clinical trials and be tested on humans. So even though there's ethical debate on this, there is a specific way that you have to follow to be able to get your drugs on the market. And then one that's a different type of ethical debate is falsifying data. Okay? So as a scientist or as a future scientist, this is extremely important. Okay? Falsifying data, whether it's for any reason, right? Sometimes people falsify because it'll help them get a promotion, or maybe they work for a drug company and they're just trying to push their product. Maybe they just want to become famous, whatever the reason. This has a huge, huge detriment to the scientific community. Not only does it make people in the scientific community and outside the scientific community distrust the overall scientific community, but it can actually hurt the current focus of research. Because if someone comes out and says that this is an answer to a question that everyone's been looking for, and they falsified all their data, and you don't have a lot of people able to back up their results, but maybe just believe it, then you take that for granted that that is the correct answer, and it could set you back 50 years, right? And it could end up causing you to go down a rabbit hole. This is a huge deal that everyone needs to have high morals when creating their research, and they need to give their data, not falsify data. Don't cherry pick your results, right? You give all your results, not just the ones that make your story look good, right? If you do the experiment 10 times and 9 out of the 10 times it fails, but one fails as in gives you an answer you don't want, but the one time it gives you the answer that you do want, that's the only piece of data that you share with the public, that's a problem, okay? And that's a big ethical problem, okay? So let's talk about research ethics in general now. So research ethics involves the application of fundamental ethical research ethical principles to a variety of topics involving research. Examples would include the design and implementation of research involving both animal and human experimentation, as well as scientific misconduct. So like the falsifying data falls under that. Now, the Declaration of Helsinki, which was set forth in 1964, is a set of ethical principles regarding human experimentation developed for the medical community by the WMA, so the World Medical Association. So this was the first fairly complete document that most countries kind of adopted. So the basic principles of this Declaration of Helsinki was that of utmost importance is the respect for the patient. Okay, their right to self-determination and their right to make an informed decision, which we hear very often, right? Informed consent. We hear that in hospitals a lot in the U.S. Okay, it's important that they know what they're getting themselves into in a certain research trial, let's say. Now, what's important is that the researcher's duty is solely to the patient or volunteer, meaning they care most importantly about the patient overall, even over the research. As hard as that is to think about as a researcher, right? The patient is the most important part. Okay? So patient welfare takes precedence over both scientific and societal interests. And ethical considerations will take precedence over laws and regulations. Now, very importantly, same thing that we see in the hospital system as well. If a research participant is incompetent, physically or mentally incapable of giving it consent, or is a minor, then you need to get surrogate consent from somebody else. Okay. Now, the last few slides, I just want to talk about what is, right versus wrong. All right. When we talk about ethics, you talk about right versus wrong. And the ultimate debate, right? Ethics is the ultimate debate of right versus wrong. And there is no correct answer. There may be one that you favor slightly, but there's no truly correct answer when we're talking about this. And the question is going to be asked forever. So much of modern biotech has a markedly lifespan and health span focused approach. So lifespan, making people live from, say, not just 80 years old to 100 years old. Health span, actually being healthy till you're 80, instead of 70, say, something like that. Now, while that is a morally, good, morally just goal, there's still, lingering fears that exist that scientists have too much control over human evolution and destiny, right? Because the better we get with technology and biotechnology specifically, the more control we have over the human body and the animals and plants and bacteria around us, right? And it's that fear of, we are becoming a godlike figure. We can affect destiny. Okay? So in addition to laws and regulations, both public and political opinions can affect the current and future state of research and development. And this is something that we're forever going to have to work with, right? That's not going away, right? Scientists can't just stay in a hole and do their research without worrying or taking into consideration what politicians and the public have to say about it, okay? For better or worse. So let's talk about a specific aspect, right? We'll bring up Dolly the sheep, if any of us remember that. Okay, this happened in 1996 in Scotland. Dolly was the first animal to be cloned from an adult somatic cell using the process of nuclear transfer. So what that was is the nucleus from an adult cell is transferred into an unfertilized, enucleated, so without a nucleus, zygote, which is a egg cell, right? So you have an egg cell, you take out the nucleus, you take the nucleus from an adult cell from a different animal, take that nucleus out, put it into the egg. So okay, zygote, enucleated, adult cell with a nucleus. Take that out, put it into here. Okay, and then try to grow an organism. Well, they actually did this with the sheep. And that sheep lasted about 6 years before passing away. Now, this brought up a lot of questions and will continue to bring up a lot of questions in the fact of, is science unstoppable? Or can we actually limit how far we go based on rational, ethical thinking? So just because certain science is possible doesn't mean that it'll actually come to fruition if we as scientists and researchers think rationally and ethically. But the public always fears that science and scientists won't, right? It'll always be, we want to do this just because we can do it, not because we think it's going to be beneficial, right? That's why this is kind of a just because we can, should we type of a question. Okay? So what's going to happen is that it may fall upon the scientific community to start the ethical discussion within itself Okay, and then as human biotech research progresses, as it's doing right now, ethical debates will continue to heat up and will continue to be extremely hot. Okay, so not only actions, but also the intentions of the scientific community require rational reasoning and justification. So an example here for our ethics is whole genome sequencing. All right, pretty expensive in general, but it's becoming cheaper and cheaper. And we have that $1,000 genome in sight, meaning I might in the near future be able to send in a sample of my DNA and for only $1,000 get my entire genetic profile, right? Which would be amazing. But whole genome sequencing differs from current prenatal genetic testing, okay? First of all, current tests identify only serious genetic conditions in fetuses, and they have a moderate to high risk. Whereas prenatal whole genome sequencing could allow for the entire genetic profile, and this could lead to the possibility of wanting to make that Build-A-Bear type kid, right? You know, I can see that my kid will have brown eyes. Well, maybe we can do some gene therapy and give him or her blue eyes. Or maybe it's my child is going to be a boy. Is it early enough to where I can change it to be a girl? Or my child will have autism or another type of disease that is not considered seriously fatal, right? That's not currently being tested. I think we only test for somewhere, I could be wrong, I think around like 13 to 15 different genetic conditions. But this could lead to a situation where, you know, maybe my child will have achondroplasia, right? So might be a little person. You know, any of these things that aren't perfectly in line with what the parents wanted, they could either fix that to their liking or decide to not proceed with the pregnancy. You know, so a lot of these ethical issues will come up. Okay, also this whole genome sequencing, as I said before, you can get your whole genome profile, but maybe the insurance companies, right, the health insurance companies get a hold of that. They would be able to possibly then decline you based on your risk of a certain disease. Okay, big ethics problem. Okay, but on the good side, golden rice, a genetically modified organism. This is a rice strain that was modified to contain beta-carotene. Okay, beta-carotene is a precursor of vitamin A. Okay, a single serving of this golden rice supplies 60% of the recommended dietary allowance of vitamin A. Now, this is important because according to the World Health Organization, more than 250 million preschool-aged children are vitamin A deficient. And over 2 million deaths and more than 500,000 cases of blindness are due to vitamin A deficiency worldwide. So just the introduction of this strain of rice can help decrease this. And in fact, there have been estimates that, you know, over 1 million people have been saved, you know, from dying in the several years that the golden rice has been on the market. And it will just continue to rise as year by year goes by. So this is a good aspect. However, still going to be ethical debates, right? Because there's always going to be people who are for or against genetically modified organisms. Now, I have just last 2 slides, and I want to leave you with some questions to ponder. So when thinking about research, right, any specific research, right, you want to know, what's the societal impact? Are any human rights being violated, right? That's a big one. Do the benefits outweigh the risks? And do any means justify a good end, right? So the means justify the end, right? Or sorry, does the end justify the means? But does any means, how you get to that result, is that okay? Does biotech ultimately advance or impede the common good of humanity and the world as a whole? And this is probably the toughest one to think about. Is there anything that we haven't thought about yet? Okay, that's the toughest one to think about because you think about everything, right? And then there's always something later. So this one you really have to prepare, right? Prepare and think. Okay. So how do you answer questions on ethical debates? Well, the biggest thing that I could tell you is do your own research, right? Do your own literature research. You should be the expert on all the experiments that have been done before, right? Both for and against, right? You want to know both sides of the argument, why it's an ethical debate fine, why it's bad, right? You need to do your literature research. That's a huge, huge thing. You need to understand the science and you need to understand the ethical ramifications from this, okay? Now, this is extremely difficult, okay? Even for people in the scientific community, literature analysis, right, is a tough job, right? It's tough to do. You get better every time you do it, but it's really tough, okay? It's a tough skill to master. And for those of you outside of the scientific community, it's even harder because they aren't as strong in the subjects of biology, of genetics, of biotechnology, right? They don't understand it, right? So they might not understand why you would use a certain experiment or why you might have to use animal experimentation, you know? And a lot of the disagreements come from the fact that many people are uninformed or many people don't understand the science, okay? And the media, right? The scientists aren't usually the ones talking straight to the public. The media are the ones talking to the public and the politicians, right? The media, as much as we want them to know everything, right? They can't be expected to understand published scientific data on their own, okay? That is going to land on the scientific community. We need to educate the general public and especially those in the media who cover scientific topics, okay? It falls on our shoulders. We need to educate the media and we need to educate the public so we can bring them up onto the knowledge level to understand much easier. But once we educate the public, and especially once we educate the media is responsible for taking it upon themselves. I'm going to highlight that. Taking it upon themselves to gauge their level of understanding before writing articles that will reach the masses and possibly promote fear-mongering, okay? The media needs to understand the science before writing an article that could cause a huge problem in the non-scientific community, okay? And we as scientists can only do our best to educate the community, to educate the media. And then the media has to take it upon themselves that if they don't really understand what's going on, they need to reach out and do their own homework and get in touch with the scientists to prepare themselves. So that's the end of the lecture, the end of the course. I wanted to say from myself, thank you so much for joining the course. I really hope you enjoyed it. And from educator.com, we appreciate you seeing this course. Please check out some other courses. There are plenty from the high school through the college level. And, from me and educator.com, thanks for joining, and we hope to see you again.